Yes, listen. Yes, what is virus? How would you define a virus? How virus is different from bacteria, fungi, and parasite? Parasite, yes. Yes?
Virus are
Non-living.
Yes.
So Mr. Raman saying it's a non-living. Observe. Okay? Yes, define it. Listen, you are saying non-living. Yes. If it is non-living, why you are studying in living subjects? Not considered to be alive. Okay, then I can start the lecture from here. How would you define something is live and something is dead? Growth. So Mr. Raman saying growth. Growth is for living objects. Next. So there is no growth in dead. Next.
Locomotion.
Movement. Okay. So movement. So there is no movement in dead materials. Okay. So movement in a sense of what? Plants. Plants don't move, so they are dead. Then what?
They grow.
Okay. This teacher saying metabolism. So metabolism is not there in dead. Okay. Anyone? You want that?
Reproduction.
Reproduction. So reproduction present in living and here it's not there. Anyone? Anyone you want that? Vitals, maybe vitals and temperature. Vitals, temperature, all vitals, right? All these things are present to the living organisms. Now, do a virus grow? It does not grow, right? Okay, so you are saying it is a non-living, right? Did a virus has a movement?
Yes.
Movement. Does a virus move?
Yes.
Yes. Then why you said virus is non-living? Okay, let us see. Then what is metabolism? Does a virus has metabolism?
Yes. Yes.
Yes.
Yes.
Really?
Yes.
Virus don't have metabolism. Then how it is living? Listen, there is one particular property of virus that makes it a living organism. That property is reproduction. And reproduction is not a right word.
Replication.
Okay. The proper term is
Replication.
Without saying I understood. Yeah, replication. Very good. Due to the property of replication, still the virus is alive. It's a living organism. Now there are some contradictions. Virus is not a self-living body. Okay. Virus always needs a living object for its survival. Right? Virus always needs a living object for its survival. That means virus cannot infect a dead body. Virus need a living organisms for its survival. Am I correct? So, still if you have doubts, I have a very good analogy for this. Try to understand. So, imagine we are going to make a new phone, a new smartphone. Maybe we can say Vamsi phone, okay, new company, for example. So, but to make a Vamsi phone, what we need? We need a industry, a factory, where the factory will make a battery, frame, processors, displays, everything. Then we need to assemble them. So maybe phone cost 10,000, but the factory will cost 10 crores. The infrastructure is very costly. Am I correct? So first of all, I need to make a factory. Then phone is nothing. Am I correct? Do I have 10 crores now? I don't have 10 crores. How to make a phone without 10 crore investment? That is my problem. Okay? Now what I will do was, now I saw that Diksha has a factory. She has an iPhone factory. She is very super billionaire. She has 1,000 crores and she has a factory. Okay? And she is making an iPhone. Okay. Now, the idea was I will hijack her systems, her computers, because though her computers are, following instructions to assemble an iPhone, right? Now, what I will do, rather than Apple logo here, I will keep a tomato logo. So, Vamsi tomato phone, for example. Okay. So, in her robot, whatever robot that is printing Apple logo, that should be replaced by tomato. What should I do? Already she has a equipment. The printer is ready to print. Just I need to give a software command to change the print, the mechanism. Am I correct? So I need not to do anything here. I need not to have 1,000 crores. Rather than I need a new software. I will insert this software into a system where the system will, do the mechanical work. Okay. I will replace Apple logo with tomato logo. That's it. Now the phone became mine. Now her company started producing tomato phones and I can use them. Right? I hijacked her own systems and I got a new product. Isn't it a clever approach? Very clever, right? This is the same thing done by a virus. Virus don't have its own mechanism to make virus don't have a factory, a cell to make all the proteins. Now virus will go to a factory that is a cell. In that cell it will introduce viral genome. What cell will do? Cell will start taking the commands of nucleus and it will produce the proteins. In this case the virus DNA has been introduced into the cell. Now the cell will produce viral proteins. Okay, so virus is one of the smartest hijacker. It hijacked the cellular machinery. Now it is producing its own proteins. Right? Yeah, that is the virus. That's why viruses are broadly classified into two types. They are. So first I will write a viral replication. Very simple. First virus came. This is a cell. Virus came. This is called attachment. After attachment, virus will be uncoated and virus will release its DNA. Either it's DNA or RNA. DNA or RNA. Now, if it is a DNA, this DNA will be integrated into the nucleus of the cell. If it is an RNA, RNA will be directly integrated into the ribosome. Okay? Now, DNA or RNA release. Now they went into the genome factory. Now cells started making proteins. This is called as biosynthesis. Biosynthesis. Now these proteins will assemble. These proteins will assemble and they will make a baby virus. A baby virus, right? Assembly of proteins. Now this virus will be, these are called small viruses. Now this virus will be come outside in two mechanisms. If the virus coming by gently poking the cell wall, this is called budding. If virus destroying the cell wall and coming outside, this is called as lytic cycle, lysis. Okay. Now, via budding we will get enveloped viruses. Via lysis we will get non-enveloped viruses. Okay. I will discuss that later. Now listen. If it is a DNA virus, viruses are broadly classified into two types, DNA or RNA. See, either virus will be a viral DNA virus or it will be a RNA virus, but both DNA and RNA cannot exist in a single virus. Either virus will have DNA genome or it will have a RNA genome, but not both. This is one important criteria. Either it is a DNA or it is a RNA. Let us say if it is a DNA virus, the genome will be introduced into the nucleus. If it is a RNA, genome will be introduced into the cytoplasm of the cell. Is it clear? And that is how it will start hijacking the cellular machinery. From cytoplasm, ribosome directly will produce proteins. In case of DNA, nucleus through mRNA and ribosome. Okay. It will take a DNA then wrap. Okay. DNA to RNA and RNA to ribosome and protein. Is it clear?
Yes.
Now, viruses are specific in their location of infection. Because, we cannot say as a virus, let us assume, we got a type of, actually that is the weakness of a cell. The weakness of a cell was cell ribosomes always listen the commands given by the nucleus. Am I correct? If nucleus said prepare HB protein, the ribosome will prepare a HB protein. So cell is always under the control of nucleus. Now virus has hijacked the nucleus, the information system, and now it's protein its own proteins, right? That means these viruses are nothing but an escaped proteins that got a good survival mechanism. These are some escaped instructions of information. If this information landing in a cell automatically preparing a protein. So viruses are the outcome of living actually. Viruses came after living organisms came. So viruses are nothing but escaped genomes and escaped proteins working like their own entities. Okay, accidentally viruses came and now they evolved to be more specific. Okay, they are just escaped proteins, escaped genetic materials that got a good survival mechanism. Survival mechanism in a sense, if they are landing in another cell, they are doing the same process. So they got that pathway, proper right pathway. Now, that is why viruses took a long time to adopt to a particular cell. If a virus that understood the mechanism of liver, it will only attack the liver, it cannot attack the other cells because it understood the liver mechanism, liver cell mechanism. If the virus that is attacking your brain, it will only attack brain because that is what it understood for past millions of years. Because that is only escaped proteins of brain, not liver. And liver escaped proteins become liver specific viruses. So always viruses have a specific location for the infection. Is it clear? That's why viruses are specific in their this one. And viruses are always obligate parasites. There is no such as what is other one? Facultative. There are no friendly viruses. All viruses are infectious. Right? Yeah. Then coming to the classification. Viruses are broadly classified into DNA viruses and RNA viruses. This is very important. Now, I can confidently say that what is a coronavirus? Is it a DNA or RNA? Most of you don't know the answer. Is COVID a DNA or RNA virus? HIV. Is HIV a DNA or RNA? Yes or no? HIV. Is it a DNA virus or RNA virus? See, still there are confusions. Chickenpox, DNA or RNA? Measles, DNA RNA. See, polio, DNA RNA. Yes.
Yes.
That's why classification matters a lot. I will let you know why. Why we need to memorize classification. Listen, if it is a DNA virus, we need to do something called PCR. In PCR, directly we have a step. First, DNA is a DNA unwinding, then primer attachment, then primer extension. That is the normal protocol of PCR. That can be directly done for a DNA virus. Take a DNA virus, it has a genome, go for regular PCR and you will get a viral genome amplification. What if it is an RNA virus? Can you able to directly do a PCR with RNA? It's not possible. You understood? The first step in PCR is denaturation at the highest temperature, 72 degrees centigrade. At 72 degrees centigrade, DNA will be destroyed. That means RNA will be destroyed. DNA unwinded. That means RNA will directly destroyed at these high temperatures. Got it? Then what to do in this case? How to replicate an RNA genome? You cannot use a regular DNA PCR for the amplification of RNA genome. Then what should you do? There is no RNA PCR. Yeah. Any guesses? Now, Then we need to convert RNA to a DNA, and DNA will be subjected to PCR technique. What is this? How to convert an RNA to DNA? Very simple. Take a RNA strand. Now use an enzyme called reverse transcriptase. Reverse transcriptase will make a complementary strand of this particular RNA. Now this became a DNA, double-stranded RNA. This is called as double-stranded RNA. This is actually looks like DNA, but it's RNA that has double strands. That means today you understood that RNA need not to be a single strand. RNA can be also in double strand. Double-stranded RNA. Got it? So simple thing, take RNA genome, incubate it in a test tube for half an hour in the presence of reverse transcriptase enzyme. Reverse transcriptase made a DNA structure. Now this DNA will be subjected to normal PCR steps. This PCR is called as RT-PCR, reverse transcriptase PCR. RT-PCR is used for amplification of RNA. So, COVID, coronavirus, is it a DNA virus or RNA virus? It is an RNA virus. So, what type of method you should know? RT-PCR or normal PCR? RT-PCR. And what is the mandatory step in RT-PCR? The first step, incubation of genome in reverse transcriptase enzyme. Without this step, you cannot go for RT-PCR. So that is why we must know what are DNA viruses and what are RNA viruses. Okay? Yeah. What is the time? How much time? 12:50, right? Lunch time. Okay, I can take one slowly there. No worries. Now listen. Classification of viruses. So viruses are broadly classified into two categories: DNA and RNA. Now each virus we have kept a families to the viruses. So what do I mean by families to a virus? Understanding the family will give better insights about the organism. I will give you one example. There is a family called primates. What animals present in primates? Yes. What animals present in primates family? Monkeys, baboons, all monkey-like things, including humans, are kept in primate family. Whose head birds? Someone saying something. Okay. Then in the family of canine, what are present? What animals present in the family of canine?
Lion.
Dogs, foxes, all these are canine family. Okay. Wolves, all are canine. Then what animals present in feline? Cats, very good. Including lions, tigers, panthers, leopards, everything are under cats family, that is feline. So see, so tomorrow if I say, I saw an animal that's looking like a feline, so you can automatically understand its features. Maybe it has a round face, maybe it might be strength and it might be non-vegetarian. It might have a cattish behavior because it's a feline. And if I say primates, definitely you will understand it might have 2 hands and legs. It might stand sometimes, and it might hang with some plants or trees. So understanding families will give some broad idea about the creation. Correct? So similarly, first we have given families to the viruses. Write down the families, very important. This lecture is very important. This will be the foundation for your regular classes. And don't judge the faculty, okay? Like what is the call like this, actually not teaching like this, okay? That's it. Now listen, families of viruses. Write down. I will give you some best mnemonics. Mnemonics are the best way of memorizing things. Now listen. Viruses are having DNA and RNA. Now families. Write down the important thing. DNA mnemonic. DNA mnemonic is HAP-Q. HAP3. families, PROCBT, 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 okay, PROCBT. These are the families, keep in mind. Okay. Is everything clear so far? Now listen. So you must one of these two. Okay. If none of the test two this is available, please by heart it and this will be really helpful. Okay. Now listen. In both the cases, there is one supervillain. In both RNA and DNA, there is one supervillains, two supervillains. These supervillains came by a starting letter called H. This is our supervillain. Supervillain in DNA is a hepatitis B. Is the supervillain or the worst lethal virus. The most lethal virus in DNA is hepatitis B. Okay. Then there is a RNA that also there is a supervillain starting with S, H, and Shukbir will tell the answer now. What virus belongs to H? HIV. Very good. Yeah, this is a supervillain in yes. RNA. Is it clear? I think first or second semester, whatever example will be found, Shukbir is always HIV. Anyway, listen. Okay. Now let us see what are these activities. DNA families. Write down the DNA families. H first stands for herpes viruses. Herpes viruses. Herpes viruses. Then A stands for adenoviruses. Adenoviruses. Listen, families are always end with viridae. Viridae means family. Herpes viridae. Adenoviridae. Is it clear? Families always starts with viridae. I'm not listening. Okay. Adenoviridae. Next one. I got one more point to say to you. I forgot. Sorry. If a virus present inside a cell, that is called as virus. If it is present outside the cell, it is called as, any idea? Outside the cell, virus don't have any function. It's a dead material, as you say. That is why we have given another term. If virus present outside, that is called as virion. Virion means outside the cell, virus presenting outside is called as virion. If it is present inside the cell, it is called as virus. This is new term I given to you. Back to this. I said PQ. By this you might understand, right? There might be 3 P's. Right? Write down the families are Poxviridae, Papoviruses, Papoviridae, you know that region somewhere around the world, and Parvoviridae. Yeah.
Sir,
We might be like this. If Palak is the first patient to get, we might got a Palakviridae. You know, that is how they got these names. Yeah. Pay attention here, Miss Palak, because you are not seeing the board. Pay attention. Okay?
what is the last birthday?
These are the families of DNA. What are the virus families? Herpesviridae, Adenoviridae, Poxviridae, Parvo, and Papoviridae.
Sorry, sir. Second one.
Parvo, Papovo, and Poxviridae.
Parvo.
P-A-R-V-O, Parvoviridae, Papoviridae, Papavo.
Papa.
P-P-A-V-A-V-O, Papoviridae. I will discuss later, don't worry. Next, proximity. Okay, proximity. What is this? Write down here. DNA viruses, RNA families, Paramixoviridae, Keep in mind, mixo means mucous membranes. Mixo means mucous membranes. Now you might understand what virus it might be. Can you guess what virus might be in mixo families? lung infection causing viruses. Okay, influenza. There are multiple. I will discuss. Paramyxo. Then second one. Rhabdomylidae. Rhabdo. Then Retromylidae. Rhabdomylidae. Retromylidae. Then next one. Orthomyxomylidae. Orthomyxomylidae. Then C. Yeah, your favorite celebrity family. C. Can anyone guess what is that family? There should be. There is an entire family for this actually. And we know this family since 1970s. Since 1970s we know all these families by the way. Can you guess what is that family? Coronaviridae. Coronaviridae is not a new virus actually. It's one of the old family. And we didn't expect it as corona will be a celebrity in 2020. Coronaviridae, then PT. PT means Bunyaviridae and Palmaviridae. Bunya, yes, it's a region in somewhere in Africa. Bunyaviridae and the Togaviridae. Togaviridae. These are the families of viruses. First of all, memorize the families. Okay? A quick recall. DNA assume herpesviridae, adrenomylidae, pox, carbo, papago, then paramyxo, retro, rhabdo, orthomyxo, corona, bunya, toga. Okay. Now, listen. What are the viruses present in HACU in these families? For viruses, I have another mnemonic, very simple to memorize. Very simple. The viruses present in HACU. Write down. The mnemonic is life on planet Earth is possible because of what molecule? Life on planet Earth is possible because of this particular molecule. What is that molecule? What is the molecule of life? What? Rain, water. I said biomolecule. Yes, Palak. Yes. DNA. Very sad. DNA has the potential. Okay? DNA has the potential to make life on planet Earth. Am I correct? So the mnemonic is, keep in mind, not DNA by the way. The mnemonic is DNA has the potential. Has is the mnemonic. DNA has the potential. DNA has the potential. Has is my mnemonic. HAS. For viruses, write down the viruses. H. Hepatitis. Hepatitis. What hepatitis? Hepatitis B. Keep in mind, only hepatitis B is DNA. A and C are RNA. Hepatitis A, hepatitis C are RNA viruses. Only hepatitis B is DNA virus. Keep in mind. Hepatitis B is DNA virus. Okay. Hepatitis B. And next one, I just want to elaborate here. Herpes viruses. Herpes viruses themselves have 3 subfamilies. That is my problem. That's why I didn't say it here. Herpes has 3 families, or you will remember from today onwards. All herpes viruses are sexually transmitted viral infections. Okay? Herpes viruses are one of the baddest viruses. They are sexually transmitted. They will cause cancers and they will cause fetal death. If a mother catches a herpes virus infection, the fetus will die. That's it. Once delivered, fetus will die within a week. Such viruses are called as teratogenic viruses. Very, very saddest thing. Reason, the problem was with teratogenic viruses, mother won't show any symptoms. If mother has a herpes infection, she won't even get a fever also. But the fetus will die inside the stomach. down. Herpes simplex 1, herpes simplex 2, Epstein-Barr virus. These are the alpha herpes viruses. Herpes simplex 1, 2 and Epstein-Barr virus. Next one, beta. Epstein-Barr virus, EBV, Epstein-Barr virus. Epstein-Barr virus. Then, okay, Epstein-Barr virus is very simple. You are one type of chicken pox. It's not chicken pox, but it is one type of shingles. Whenever shingles, it's the chicken pox in old people. That is very deadly. If old people got chicken pox second time, they might die. Chances of that is very high. That is also called as varicella zoster. This relapse of EBV is called as varicella zoster. That I will discuss later. Then second one, beta family. Beta family has a virus called CMV, cytomegalovirus. So what will cause to this cytomegalovirus? If you get CMV infection, your brain will dead. You will go to coma and you will die. Because this virus will attack the brain. Okay? So beta virus is CMV, cytomegalovirus. And last and final, gamma virus is our varicella zoster. Varicella zoster. These are the viruses belongs to herpes virulence. Okay? Next one, HAS. Then A stands for adenovirus, directly adenovirus. So what is adenovirus infection? Can anyone guess what is adenoid gland? Where is adenoid gland?
Respiratory.
It's in the nasopharynx, right? So after getting this viral infection, your adenoid gland will be inflamed. It will become swollen. That's why this virus is called adenovirus. So adenovirus is a DNA virus, keep in mind. Yes, this what is right?
CZ.
Where is that right?
CZ.
BCZ. See, actually, you know, first year so many internationals. Now, even I got same problem with you in the first semester. Right? Now I became back to that old toxic. Anyway, this one, yes. S stands for smallpox. Smallpox. Okay, smallpox virus. And last and final, our serial killer. That is always a hepatitis B. These are the viruses of DNA. So a quick recall. Herpes viruses, 3 families, alpha, beta, gamma. Herpes simplex 1, herpes simplex 2, Epstein-Barr virus. Beta family, after B there is a C in alphabets. So cytomegalovirus. Then gamma, varicella zoster. Then you have smallpox and adenovirus. Then coming to the RNA virus pneumonia. RNA virus is pneumonia. Right now, very simple pneumonia. PCR square, MMR square. PCR square, MMR square. PCR square, MMR square. So now write down the virus names. PCR square and MMR square is the RNA viruses. Write down the names. P. First one P. Poliovirus. Poliovirus. Then C. Yes, celebrity virus. Tell me now. Any guess?
Coronavirus.
Very good. Coronavirus. No, coronavirus. Then R. R square. That means 2 R's. Write down. Rhinovirus. Rhinovirus. And next one is rotavirus. Rotavirus. I want to tell about rhinovirus. Listen. Do you know the difference between rhinoceros and hippopotamus? How hippo is different from rhino? Yes. If you still don't know, there is a cement company called something rhino, right? Yes. Tell me, Mrs. Rhino has what? It has a very big nose. That nose horn. A horn nose, right? That means rhinovirus will cause nose infections. Common cold. Common cold is caused by rhinoviruses. Now you understood, right? Why they call as rhino. Rhino means nose, by the way, in Greek. Okay, rhino, rhino. They kept same animal. or line. Okay? Yeah. Next one, rotavirus. Rotavirus, the morphology looks like a ball, like a circle. Circle is called as a rotation, rota. So that is why it is called rotavirus. Rotavirus will cause virus-mediated dysentery and motions. Virus-mediated, those motions will be given by rotavirus. Next one. MMR spray. You already know these viruses because you are getting these vaccines. What are those? Tell me. M. Measles. Mumps. Yes, Mr. Chuma, right? Yes, your name is Chuma. Somewhere in this.
Yes, sir.
Yes. I agree. Measles, mumps, then R spray. Tell me, what are those? Rabies. Rabies virus and last and final. Rotavirus. Yes. Retrovirus.
Measles.
Retrovirus has some interesting features. Reason, very, very interesting. It's an RNA virus, but RNA will be converted to DNA and DNA will be kept inside the human cell. Keep in mind, it's an RNA virus. RNA virus has a enzyme. It will make a pseudo DNA. Pseudo DNA will be kept in our DNA. It looks like a DNA virus, but it's an RNA. It will do reverse game. RNA to DNA. Usually it is like this, DNA to RNA, then RNA to proteins. In this case, a virus, a RNA virus making a DNA and keeping DNA in the nucleus. It's a reverse. What virus is this? Yes, Kushi. What? I will let you know. Listen, this particular virus is one of the most challenging virus infection to treat. The reason is, listen, if DNA is converted to RNA, the rate of mutation is very less. Every 1,000 copies, one error will happen from DNA to RNA conversion. Got it? Why evolution favored DNA to RNA? Because the rate of mutation is very less. But if you are trying to convert an RNA to DNA, the rate of mistakes are very high. 500 mistakes for every 1,000 copies. Now one virus took this advantage. Let me convert my RNA to DNA so I will get more variations so the host cannot detect me. The host immune system cannot detect me. Okay. Then next thing, the first priority is more mutations in the generations. Second priority, it is attacking the immune cell, that is helper T cell. Now you can guess what is this virus. This is HIV. HIV will convert the RNA to DNA and the DNA will be inserted in our cell. Now our body cannot detect like HIV because now it is hiding as a DNA in the cell. Until the patient's immune system drops, DNA will be converted to RNA and it will lead to AIDS condition and AIDS will lead to death. So yes, HIV is one of the challenging viruses because it is having this pleomorphism. Pleomorphism is various forms of this virus and it is doing this reverse game. That's why this was kept in a family called Retroviridae. Retro means reverse game. Reverse game. Yeah, these are the viruses of RNA. So tomorrow if I ask what is polio, is it DNA or RNA? Polio is
RNA.
RNA. Then hepatitis B,
DNA.
smallpox, DNA. Smallpox is DNA. Okay. Corona,
RNA.
so now you got some idea, right? Classification of viruses. Try to practice and it will be easy for you. Yes? Kadam B. Okay. Thank you, class. Let me take your attendance now. Yes, thank you so much. After a long time I am coming here.